Good afternoon, everyone. Here I am Tribeni Hazarika, a research scholar from the Department of Zoology of Arunachal University of Studies. Today, I will be going to discuss on a very interesting topic of cell biology, that is the cell cycle and cell cycle checkpoints. So, before going to the topic in detail, let us first know the learning outcome of the topic. So, there are different types of, there are different aspects of cell cycle which will help us in many ways. First of all, it will help us to understand the necessity of the cell cycle, then to summarize the different phases of the cell cycle, then to understand the regulation of the cell cycle, and to understand the significance of the cell cycle and checkpoints. As we all know that cell divide in order to grow and repair tissue, and it reproduces themselves. So the definition of cell cycle is written like this here. The cell cycle is the sequence of events by which cell duplicates its genome, synthesis the other constituent of the cell, and eventually divides into two daughter cells. What does this sentence mean? This means that the cell cycle is achieved by duplicating their contents and by dividing into two, in which each parental cell give rise to two daughter cell each time they divide. And also we can say that cycle of the cell growth and division allows a single cell to form a structure consisting of millions of cells. The cell cycle division is the fundamental means by which all living things are propagated. In unicellular organism, each cell division produces an additional organism, whereas in multicellular organism, repeated cycle of cell growth and division are required. So thus the cell cycle consists of four coordinated process, that is the cell growth, DNA replication, distribution of the duplicated chromosomes to daughter cells, and cell division. So this is all about the cell cycle and its necessity in brief. And now I'll be going to discuss about the different phases of the cell cycle. So the concept of the cell cycle and its different phases was first discussed by Alma Howard in collaboration with Stephen Pelk, a physicist, in the year 1953. According to Howard and Pelk, there are four phases of cell cycle. The first one is the G1 phase, S phase, G2 phase, and M phase. And if we observe under a microscope, the cell cycle consists of two basic parts, the non-dividing part called the interphase, which consists of 95% of the cell cycle, and it includes the events of the G1 phase, the S phase, and G2 phase. And the dividing part includes all the events of M phase, that is the mitotic phase. This prophase, metaphase, anaphase, and telophase. So, although the interphase is also called as the resting phase, but this is the time when the cell is preparing for division by undergoing cell growth and DNA replication in an orderly manner. And the key molecular events that takes place during the cell cycle are DNA replication or DNA synthesis and the chromosome segregation. And the DNA replication or DNA synthesis occurs in the S phase here, and the chromosome segregation, which is a result of mitosis, occurs during the M phase of the cell cycle. It is the mitosis phase. And it is mentioned that a typical mammalian cell divides approximately every 24 hours, where G1 phase lasts for 11 hours, S phase lasts for 8 hours, G2 phase lasts for 4 hours, and M phase is the shortest, which lasts for only about 1 hour. But there are certain cells also which which rapidly divides. For example, each cell take only 90 minutes for budding, and there are certain other cells which cease proliferation and enter into a quiescent state called the G0 phase. This is the G0 phase, and the cells present in the G0 phase is called as the resting cells, or are called the resting cells. So this is about the phases of the cell cycle in short. Now I'll be going to discuss the different phases of the cell cycle in more detail. G1 phase or the gap 1 phase occupies 30 to 50% time of the cell cycle. And it is important to know that during this phase, though the cell is metabolically active and the cell continues to grow, but there is no DNA replication. It involves or it increases the production of different types of RNA like messenger RNA, transfer RNA, ribosomal RNA, and it also involves the synthesis of proteins and cell organelles which lead to the growth of the daughter cells. The proteins synthesized during G1 phase are the regulatory protein which control various events of mitosis. This regulatory protein also play a very important role during the regulation of the cell cycle. And during the S phase, there are various enzymes which also helps in the DNA synthesis, such as DNA polymerase. And a very important protein called as tubulin, which helps in the formation of spindle fibers or the mitotic spindle, are also synthesized during the G1 phase. So this is about the G1 phase. Then S phase, which is also known as the synthesis phase, it occupies 35 to 45% of the cell cycle, as it is mentioned here. And during this phase, replication of DNA and synthesis of histone proteins occurs. And the most important part of S phase is the sister chromatid cohesion. During the S phase, actually what happens, the sister chromatids are held together by the process of cohesion with the help of a complex protein called cohesion. And the state of the cohesion plays a very important role as it links the sister chromatids together, and this cohesion ring is destroyed during the chromosome segregation, which takes place during the M phase of the cell cycle. And at the end of the S phase, the chromosome contains two DNA molecules and duplicate set of genes. That is, the amount of DNA per cell doubles at the end of S phase. So this is about the S phase. During G2 phase or the gap 2 phase, it occupies 10 to 20% of the cell cycle. This phase is the period of rapid cell growth and protein synthesis, during which the cell prepares for mitosis and cell division. And the G2 phase also helps the microtubules to reorganize in the formation of the mitotic spindle. M phase or the mitotic phase. Actually, in this phase, the M phase represents the phase when the actual cell division or mitosis occurs. Mitosis starts at the culmination point of the non-dividing part of the cell, that is the interphase. And this mitosis is divided into 4 phases. First one is the prophase, metaphase, anaphase, and telophase. Here is the image showing different phases of mitosis. As you can see here, during prophase, the chromatin condenses to form the chromosome, and during the metaphase, the chromosomes line up along the metaphase plate. Then during anaphase, the sister chromatid move towards the opposite pole, and during telophase, the nuclear membrane and nucleoli reappears and the chromosome unwind with the chromatin fibers. So this is all about the mitotic phase. As I have already mentioned in the previous slide, that many cells cease proliferation and enter a quiescent stage called G0 phase. Actually, in this phase, in multicellular organism, most differentiated cell like nerve cell, they exit the cell cycle in G1 phase and then they enter the G0 phase without the cell division during the entire lifetime. But there are certain G0 cell which can return to the cell cycle, such as the cells of the internal organs like liver, and they resume replicating. So this is all about the G0 phase. Then coming to the cell cycle regulation. As you all know that cell cycle is a continuous process inside our body. Cells are constantly receiving signals in order to proliferate or grow in terms of growth factors. Once they receive signal, the only thing it can do is that they grow and divide, but there must be some sort of regulation that will ultimately regulate how actually the cell will divide, or because if it is not regulated or beyond our regulation of growth, then there will be masses of cell, that is tumors will formed, and that tumors will gain properties that will make it malignant or cancerous in nature, which will not be good for the health of any organism. So the progression of the cell through the cell cycle is regulated by various external and internal signals or factors, and these internal signals are also called as the control points or the checkpoints. So here we can see that in each cell, the cell cycle can be regulated by various external factors like nutrient availability, mating factors, cell size. So these factors help the cell to regulate at the point of G1 phase, which is also known as the start point. In animal cell, the cell cycle is regulated by the various external or by various growth factors at the G1 phase, which is known as the restriction point. But in the absence of this growth factor, the cells enters the G0 phase. Now, what are these checkpoints, or what are the internal control units? So these checkpoints actually in each step of the cell cycle is controlled by the internal controls, which are called checkpoints. These checkpoints actually help us to know whether the preparations for the cell division are sufficient or not, whether the requirements are sufficient or not. So this helps. So this was checked by the checkpoints. The different phases of the cell cycle are coordinated with each other. This synchronization between the different phases of the cell cycle prevents the entry into the next phase of the cell cycle until the events of the preceding phase is accomplished. So going to the cell cycle, there are three cell cycle checkpoints: the G1S checkpoint, the G2M checkpoint, and M checkpoint. So these are the three cell cycle checkpoints. Now I'll be discussing the three checkpoints in more detail. So here is the image showing the three different checkpoints of the cell cycle. So this one is the G1 checkpoint, which takes place between the G1 and S phase. Then G2 phase occurs at the end of the G2 phase, and the M phase, which occurs or takes place at the end of the metaphase during the process of mitosis. The G1 checkpoint is also known as DNA damage checkpoint. Why it is also called as DNA checkpoint, I will tell it later. First of all, I would like to say that in mammalian cell, the G1 checkpoint is also known as restriction point, and in yeast cell, it is known as start point. And now the question is, why this G1 checkpoint is also called as restriction point? Because this is the time when the cell gets committed for completing the cell cycle. So for this region, the G1 checkpoint is also called as the restriction point. This G1 checkpoint help us to know or to check whether all the preparations such as enzymes, proteins, and ATP are produced sufficiently or not. And it also checks if there is any damage to DNA, because in the next phase the DNA is going to replicate, and if there is any damaged DNA, the damaged DNA will replicate. So it is going to check for the damaged DNA. So the G1 checkpoint is also called as DNA damage checkpoint. And this G1 checkpoint also activates the cycling dependent kinase enzyme, which is responsible for the initiation of transcription. Now coming to the G2 checkpoint. G2 checkpoint help us to check whether the DNA has been properly replicated or not, and whether the proteins, enzymes, and the ATP are properly synthesized or not. So this was checked by the G2M checkpoint. And if the DNA has not been properly replicated, then it prevents the initiation of mitosis if the cell contains any damaged DNA. And the G2 checkpoint also checks the whether the synthesis of a more of a most important protein called the tubulin protein, which helps in the formation of the microtubules or the spindle fibers. So this synthesis of protein was checked by the G2M checkpoint. Then the last checkpoint, that is the M phase checkpoint, is also known as the spindle checkpoint. This checkpoint takes place at the end of the metaphase, as I have already mentioned in the diagram. So this checkpoint examines the proper alignment of chromosomes on the mitotic spindle, and if the chromosomes are not properly arranged, the cell post mitosis, and it checks if the sister chromatids are properly arranged or not. Then only it continues for the mitosis process. So this is all about the cell cycle and the cell cycle checkpoints in short. So at last, I would like to thank all the authorities of the Arunachal University of Studies for giving me this opportunity for presenting this teaching video in front of you all. So thank you so much.